There are two forms of glucose that you could be expected to draw. One is alpha D-glucose and the other beta D-glucose, otherwise commonly known as alpha and beta glucose. The only difference between them is the placement of the H and the OH on the carbon furthest to the right in the ring. In the alpha glucose, you find the hydrogen at the top and the OH at the bottom, whereas in the beta glucose, you find the OH at the top and the hydrogen at the bottom. Glucose is a hexose sugar. That means it has 6 carbons. 5 of them are contained within the ring, and the 6th one is attached to the top. Like glucose, ribose is also a monosaccharide. However, this is a pentose sugar. It contains 5 carbons, 4 of which are contained within the ring, and 1 of them at the top. In a saturated fatty acid, the first carbon is part of a carboxyl group where the carbon is attached to an oxygen with a double bond and a OH. Thereafter, you have a carbon chain. It can be any number of carbons, N, and then at the end you have a carbon attached to 3 hydrogens. When there are no double bonds between the carbons, they are carrying the maximum number of hydrogens possible. We call this a saturated fatty acid. The bottom diagram in white shows you a condensed or simplified way of drawing a saturated fatty acid, where N represents any number of carbon atoms in the carbon chain. In our example, N equals 4. Here I'm going to show you the structure of an amino acid. The nitrogen is attached to 2 hydrogens. We call this an amine group. The carbon on the right hand side is attached to an oxygen with a double bond and an OH, and we've seen this before. This is a carboxyl group. And the central carbon is attached to a hydrogen, and R represents a variable group, so this can be any element or any other group that's attached to that central carbon atom. It is that R group that defines a specific amino acid.